Hi, today we are going to talk about the cellular part of the innate immunity, and this is our third lecture in the immunology course. Um, Maybe you have noticed that it is a bit dark, maybe the lighting is not so good, but it is 11 o'clock in the evening and I had to record the lecture today, so I apologize if the photos are not so good, but I'll do my best to remain focused and do the lecture as best as I can. So let's start talking about the cellular part of the innate immune system. This image we've seen before, and today we're going to talk about these cells, which are part of the cells that are part of the innate immune system. As you might still remember, we talked about the innate immunity, and we discussed that the innate immune system does not have specificity, so it cannot recognize different types of pathogens or different types of antigens. And this is why it does not have memory, and every time it reacts against a certain antigen exactly the same way, regardless of the number of times that it has encountered that antigen. However, to initiate an immune response, these immune cells, they need to have the ability to recognize or differentiate self. Sorry, let me sort out this problem. Okay, so although the immune cells, they do not have to recognize different types of antigens or shapes of antigens, but still they need to recognize or differentiate self from non-self so that they can conduct an immune reaction against the non-self. So these are the things that the immune system, even the innate immune system, which is non-specific, need to recognize to initiate an immune response. First, the immune cells need to recognize self. They need also to recognize absence of self, and they need to recognize non-self. What do we mean by that? So how do these cells, the immune cells, the innate immune cells, how do they recognize self? How do they know that this cell is part of the normal cells, of this person's cells? There must be an ID that defines this cell as a self cell. This ID is the MHC molecule, or MHC, which is an abbreviation of the major histocompatibility complex. And for human beings, we use the HLA. And this is why we have different HLA typing, and every person has certain or specific type of the HLA. And when we want to do organ donation, if there is an organ donation, we need to check that the HLA typing is matched. Otherwise, there would be a rejection of that organ because it would initiate an immune response. So this is the ID the cells use to define itself as a self. This MHC molecule is presented in all nucleated cells, so all cells should have the same MHC molecule or HLA molecule in the humans to be recognized as a self cell. The other part of the self, which we mentioned last time, is the normal flora, and we stated that normal flora is part of the innate immunity because it prevents the anchorage or the attachment of more pathogenic bacteria. Still, these normal flora, although they are not self and they are not presenting the MHC molecule, they are considered as part of the self. And as you might remember, last time we talked that this is because there's a sort of a deal between the human body or that organ with these bacteria to be recognized as self and provide them with a habitat. At the same time, in return, these bacteria will provide the human body with protection against more pathogenic bacteria. This sort of relationship is also regulated and organized, and this is regulated by a specific type of T cells, which was recently discovered, called the Treg, which is responsible for the regulation of the immune response. So this is how the normal flora, although they are not self, they are still considered as a self. This is one way, or one part that the innate immune cells need to do, recognizing self. The other part is that the innate immune cells need to recognize absence of self. So any cell that does not have the ID, the MHC1 molecule, would be recognized as non-self, or would be recognized as abnormal cell and would be attacked by the immune system. And this particularly works for the natural killer cells, as we're going to see later. And the third part is the need to recognize the non-self, recognizing structures that would never be present in a human body or in a self. For example, like broadly speaking, having feathers. Feathers should never be part of the human body, so it would always be considered as antigens. So we'll go in details in defining or presenting the way that these innate immune cells manage to recognize self from non-self. Still, as you have noticed, we did not specify different types of antigens. We are just recognizing everything as either self or non-self. While we are talking about how these immune cells conduct these functions, natural killer cells are going to be the focus of our talk in the coming few slides. So how do cells recognize self? This image here shows the healthy cell, and this is the MHC molecule, class 1 MHC molecule, because class 2, we have another class which is class 2 MHC molecule that is located and presented on immune cells. We will discuss this in the coming lectures, but this is the MHC. This is the MHC class 1, okay? And so as I said, every person have a specific MHC or HLA type, and this would give this person his specificity. Other than that, these MHC molecule, they tend to carry or hold, as you can see here. Let me just make it work this here, okay? So these MHC molecule holds or carry the self proteins. So the normal healthy cell will keep renovating or refurbishing its interiors. So by degrading old organelles and building up new organelles, the proteins formed from the degradation of the old organelles would be presented in the MHC class 1. So MHC class 1 keep changing and showing what is produced by this cell, and these proteins are part of the self protein. So when these cells come in contact with immune cells, the immune cells have been trained and have been taught to recognize self MHC molecule and to recognize self protein. So this would be given the OK or the green light for survival. So this is what happens when we have a normal healthy cell presenting the MHC class 1, and these are self proteins. This is a natural killer cell, and in the natural killer cell we have a killer inhibitory receptor and killer activating receptor, KIR for killer inhibitory receptor and KAR for killing activating receptor. So when we have a normal MHC, the self MHC holding normal self proteins, these would be recognized by the killing inhibitory receptors, and this will tip the balance toward inhibition of killing, so no killing would take place. let's see what happens when the cell is infected with a virus. So the virus will get inside to the cell. What viruses do, they hijack the DNA of the cell, and they will start producing viral proteins, the structures of the proteins. And as I said, the MHC molecule, which we see here, this is the MHC molecule, it just presents whatever inside the cell. So now it will start showing the viral peptides, because the cell has just started producing these viral proteins. So it will show these viral proteins. These viral proteins will be recognized by the T cell receptor, and now the killing T cell receptor or cytotoxic T cells will start the immune reaction, and killing of the infected cell will take place. This is for the T cells, which is part of the adaptive immune response, remember. So the viruses with time learned that this is a way of inhibition of their propagation or their spread. So they learned another technique. They decided that if they hijack the HLA or the MHC genes and prevented their expression on the surface of the cell, in this case the T cells would not see the MHC molecule or the HLA molecule, and would not see then the viral peptides, and this is how they would survive. And this is what the virus do to survive. They will succeed in escaping the T cell immune response. However, this does not affect the function of the natural killer cell. The natural killer cell has the ability to recognize absence of self. So this is, for example, tumor cell, or let's presume this is an infected cell with a virus, but this virus is one of the viruses that have learned to prevent the MHC gene expression, and now there is no MHC1 on the surface, and no viral antigens are presented. As I said, they would survive the T cell. Attack. However, natural killer cells would recognize that there is no MHC molecule, which usually interact with the natural killer cell inhibition receptors. So the natural killer activation receptors would work, and the natural killer cells would manage to recognize that there's something wrong with this cell. It does not have the ID. It's not showing its ID. So it will start the cytotoxic reaction and will kill that cell. So this is one of the, let's say, unique features for the natural killer cells that is not present in any of the immune cells. Ability to recognize absence of self, which comes very handy in viral infections and in tumor cells. Okay, and the third part is recognizing non-self. So the immune cells, the innate immune cells, need to recognize specific antigen structures that would never ever be present in the human body. For example, bacterial lipopolysaccharides or peptidoglycans. These peptides or these proteins would never be part of the human body. They can be part of any type of bacterial structure. It does not belong to a specific type of bacteria, does not belong to a specific species of bacteria. It is some sort of a general structure that is usually present in the bacterial pathogens. So the innate immune system, in order to recognize the non-self, they should have the ability to recognize particular structures that would never be present in the human body. We call these antigens pathogen-associated molecular patterns. And this is still a broad term. This is still a broad category of structures. It does not have the ability to differentiate between different types of pathogens. Also, the innate immune cells, they have the ability to recognize stress signals. So what are these stress signals? When the cell got affected with any other environmental factors like bacterial invasion or viral invasion, or maybe starvation, for example. So this would give an alarm and alert, and the cell would be in a condition we call it SOS condition. And this will turn on SOS genes. And the SOS genes are specific genes that are turned on only in case of emergency. And when these genes are turned on, this would give the signal for apoptosis, and this would give the signal for other immune cells to come and kill this cell. The stress signals, one of these stress signals are the MEK-A and MEK-B. We call these heat shock proteins, which are produced in the SOS conditions. Once the cells see these MEK-A and MEK-B proteins expressed on the surface of the cells, the immune cells would know that this cell is not normal, it's not healthy. They don't know exactly what's going on, they just hear the alarm ringing and know that this cell is programmed for death, and they might come and help the cytotoxic effect or the killing of that cell. They definitely don't know what's going inside that cell, whether there is abnormal growth, it is a tumor cells, and there is sort of dysregulation, severe dysregulation in the genome of this cell. So this would turn on the SOS. Maybe there's a virus, bacteria, whatever the signal. Maybe just starvation, and the cell is in the middle of cell division, and all of a sudden discover that it's in a condition of starvation and it should not complete this cell division. It should, it is stuck and it should die. So this would give this alarm signal or the stress signal, and would give the signal for the immune cells to attack these stressed out cells. Now, how does these immune cells, the innate immune cells, how would they know that the MEK-A and MEK-B are expressed or are shown on the surface? There must be receptors on the surface of these innate immune cells that can detect either these patterns, the pathogenic patterns, or can detect these alarm signals, the MEK-A, MEK-B. These receptors are called pattern recognition receptors. They are located on the surface of the immune cells, and they can detect these non-self patterns or can detect these stress signals. Example of the pattern recognition receptors, we have the toll-like receptors, very famous toll-like receptors. Many studies have been conducted on toll-like receptors. 2 and 4 are important for us because they are present on the skin and in the mucosa. So many studies are conducted to see the role of mutations in the toll-like receptors on creating sort of immune-mediated conditions. So let's see what's going on here. So this is, for example, tumor cell. Tumor cell is expressing the MHC molecule, but this tumor cell, there's something wrong going on in its genome. There is haphazard replication in the DNA of this tumor cell. This would give a signal. This is sort of protection mechanism. One of the mechanisms our body follows to to survive. So having the abnormal replication here would tell the cell that there's something wrong is going on, and this will turn on the SOS alert status, and the proteins would be expressed, the heat shock proteins would be expressed on the surface. Now these heat shock proteins, the MIC-A, MIC-B for example, they will interact with the killing activating receptors in the natural killer cells, and now the balance is stepped toward killing of this tumor cell, and this is what happens, killing of the tumor cell. This is an example of these recognition patterns, the toll-like receptors that we have. It's a big family of receptors, and each receptor is specific to recognize certain pattern, not a certain pathogen. So for example, toll-like receptor 2 is able to recognize peptidoglycans, which is usually present in gram-positive bacteria. Toll-like receptor 4 is recognizing the lipopolysaccharides, which is present in gram-negative bacteria. So notice we are talking about big categories of pathogens, not specific pathogens. Some of the toll-like receptors recognize single or double-stranded DNA or RNA, which can recognize viruses. Okay, so this is what we need to learn about the natural killer cells, their role in recognizing self and non-self, and the receptors that are located or present on the surface of the immune cells, of the innate immune cells, that would allow it to recognize pathogenic patterns or alarm or SOS molecules like MIC-A and MIC-B. Let's move to the second part, which is the neutrophils. So remember the neutrophils, we said it's the most abundant type of cells. Usually it stays, or it goes around in the blood. It is only present in the tissues only when there is an inflammation or infection, but it does not reside in the tissues normally, not like macrophages, for example. And remember also that neutrophils is part of the granulocyte, so it has granules inside it, and these granules are sort of a neutral, not acidic and not basic, and these granules are actually enzymes that would help in degradation and killing of the pathogen. Examples are proteinases, elastases, lysozymes, and defensin. Remember, defensin is a molecule that is responsible for killing of the bacteria by binding to the surface cell membrane and creating holes in the cell membrane. So these defensins molecules are also present as part of the granules inside the neutrophil. Neutrophils would be activated by the presence of different types of cytokines or chemokines or complement proteins that are produced from certain macrophages or from the complement system at the site of the infection. For example, C5a, the complement 5a, the lymphotoxin B, the interleukin 8, which is a chemokine. So these are the types of ingredients or proteins that are needed to activate the neutrophils. The neutrophils are moving in the blood vessel, and now they need to move to the tissue at the site of the infection. So this goes into 4 steps. First, the rolling or margination. The second is adhesion, transmigration or diaptesis, and migration. So let's talk about these steps in detail. This image shows us this is the blood vessel, this is the blood flow, and here is the perivascular space, and here's the site of the infection or inflammation. The blood is moving in a high velocity or high speed in the blood vessel. The presence of infection here would produce certain cytokines, chemokines, and complement proteins that will spread in the perivascular area and will go through the endothelial cells here to the perivascular area. These neutrophils, they have the receptors that is able to recognize these chemokines. This is example of the cytokines and complement systems. So these chemokines would attract the neutrophils to the margin of the blood vessel. So when the white blood cells are moving in a high speed, if you want to exit the highway, for example, if you want to take the right exit, you need to start slowing down, and you need to take the right lane until you move to the last right lane. Then you'll be able to go out through that exit. This is exactly the same. So here the white blood cells will start to slow down because of the interaction between these receptors and the chemokines. They would be attracted to the margin, and because it used to move in a very high speed, and all of a sudden it had to slow down, it will start rolling and jumping on the margins of the lymphocytes. Sorry, the margins of the blood vessel. And while jumping or coming in contact with these endothelial cells, other interactions would take place. For example, here the selectin, which is expressed, one of the adhesion molecules expressed on the endothelial cells, it will become in contact with the selectin ligands here. You see here the selectin ligands were not activated, but once it come in contact with these cytokines, it become activated, and now it will bind with the selectins. The selectins will provide sort of a weak interaction between the neutrophil and the endothelial cells, and adhesion would start to take place. Later on, more contact or more, let's say, tighter contact would take place when the integrins, when the integrins come in contact with the ligands here, and then the white blood cell would be able to go through these endothelial cells, and it will move to the site of inflammation. So during the process of activation and margination and migration, different adhesion molecules were expressed both on the endothelial cells and on the surface of the neutrophil. So we have selectins, we have integrins, we have the intercellular adhesion molecule, or ICAMs. These adhesion molecules are located at different locations. So for example, selectin, we have different types of selectins. We have P-selectin, which is expressed in different type cells, including the platelets, and they are important for the aggregation of the platelets at the site of injury. For example, if we need clotting at the site of injury, we need adhesion of the platelets. So the platelets would express the P-selectin. The E-selectin are mainly expressed in the endothelial cells. The L-selectin are expressed in the leukocytes, and they are important for leukocyte homing. Remember when we talked about the lymph node, and we talked that the white blood cells, neutrophils, would come through the venules, the high endothelial venules, and there's lots of adhesion molecules, and there would be communication between adhesion molecules between the leukocytes and the endothelial cells present in the high endothelial venules in the lymph nodes. So this would help the leukocyte to be attracted inside the lymph nodes. So this is an example of the selectin molecules. Integrins are usually expressed on leukocytes. So we have beta-1 integrins which are expressed in the T and B cells, and we have beta-2 integrins which are expressed in the monocytes and neutrophils. And finally, the cellular adhesion molecules, different type of cellular adhesion molecules. They are important for communication between the cells. Like when there is a contact between a macrophage and the neutrophil, there is a contact through the adhesion molecules, or between the endothelial cells and the leukocytes. There would be a communication between the ICAM, the ICAM adhesion molecules, and integrins. So ICAMs can be presented on the endothelial cells, and they can interact and communicate with the integrins that are expressed on the monocytes or neutrophils or T or B lymphocytes. So as you see, during the process of moving of the neutrophils from the middle of the blood vessel to the margin until it gets out of the blood vessel, there are different types of adhesion markers that have been expressed. Initially, selectins would be expressed, and this would allow the initial adhesion of the leukocytes. Then stronger binding would take place by the integrins, and eventually when the cellular adhesion molecule would allow the extravasation of the neutrophils into the tissues. Now the neutrophil is inside the tissues. We'll see how it can get to the site of infection. So let's see again what happens when we have a cut wound. These are the pathogens here. This would initiate different types of immune reactions. Initially, we have the macrophages, which are residing in the tissues. They would be able to recognize these pathogens and start engulfing the pathogens. Also, they will start producing cytokines and chemokines to attract adjacent lymph or white blood cells. Also, presence of the antigens and the pathogens will stimulate the complement system, and the complement cascade would start working on different complement proteins would be produced, and these would spread through the tissues and come to the blood vessels, affecting the endothelial cells, initiating an inflammatory reaction. So this is how the excuse me. This is how the neutrophils are attracted through the 4 steps after rolling, adhesion, and migration into the tissues. They would follow the chemoattractant, because there is a concentration of the chemoattractant or gradient of the chemoattractant concentration, so it will follow that gradient, moving toward higher concentration of the chemokine ingredient. This is the C5a again, which is the 5th complement protein. The C5b is here on that bacteria, creating the complex, the MAC complex, to create a whole. This is the C5a, which will act as a chemoattractant to attract these white blood cells to the site of infection. Now the neutrophil is activated, and it will start doing its job, producing cytotoxic chemicals, producing its granules, the proteases, lysozymes that would start killing the bacteria. So the bacteria has been attacked by different mechanisms. The complement cascade, the macrophages, and this is the 3rd way, which is through the neutrophils. There's a very beautiful video that you can see, and it shows the process of transmigration. Okay, so how does the neutrophils move from the blood vessel to the tissue? How does it go, or get the directions toward the pathogen or toward the site of infection or inflammation? This process is called chemotaxis, and this happens through the chemoattractants. So the complement components, the cytokines, the chemokines that are present at the site of infection, these would act as chemoattractants, and because there's receptors on the surface of the neutrophils that can recognize these chemoattractants and bind to it, and the neutrophils would move through that ingredient, moving to the higher concentration until it gets to the site of infection or site of inflammation. Opsonization, we've talked about it before, which is coating of the microorganism with an opsonin, and we discussed different types of opsonins. For example, C3b is an opsonin, C-reactive protein and antibodies. All of these act as opsonins, and they would act as an attractant. It will attract the macrophages to engulf this pathogen, or attract the neutrophils to attack that pathogen. So here, this shows different types of opsonins. There is antibodies. There are these complement proteins. The neutrophil has receptors for everything. It can recognize antibodies, it can recognize antibody-antigen complexes, and it can recognize opsonized pathogens through the complement, opsonized by the complement proteins. So the neutrophils is well prepared, having all of these receptors ready to recognize these opsonins and to be attracted to the site of inflammation. The third or the last step is the phagocytosis. So once the neutrophil got to the site of the infection, it was able to recognize the pathogen now. It would either release the lysozymes, the granules that it has, to attack that pathogen, or most commonly what happens is the process of phagocytosis. So the neutrophils would start engulfing the pathogen by forming pseudopodia. So these pseudopods would surround the pathogen and will form a phagosome, and this phagosome would come in contact with the lysosome that has the lysozymes, that has the proteases, and will form a phagolysosome, and this would initiate the degradation of the pathogen. To appreciate the importance of the neutrophils in the immune reaction, we'll talk about this condition, the leukocyte adhesion deficiency syndrome. This is a rare condition. It happens 1 in 100,000 person. It's an autosomal recessive condition. The problem with this disease that there is an absence in one of the complement receptor proteins. So the neutrophils are fine, the complement system is working fine, the rest of the immune cells are okay. The only problem that the neutrophil, it is missing one of the receptors that is able to recognize the complement, the third complement protein. So it would not be able to recognize that complement system. Because of that, the process of chemotaxis is not good, because there is no attraction from the neutrophil toward that complement protein, because there is no receptor on the neutrophils. For this problem, that person has a defective immune response, and whenever there is a bacterial infection, usually there is no abscess formed, because there is no neutrophils, no adequate number of neutrophils are located or are present at the site of infection. Usually those patients should be treated with a bone marrow transplant to get a normal neutrophils, to get progenitor cells of normal neutrophils. This image shows an example of a case, a reported case of a leukocyte adhesion deficiency. This is a child, and this child has different skin infections, and what is peculiar about these skin infections that there is no abscesses formed. So there is a site of infection, there is inflammation, but there is no pus is formed at this bacterial infection. Also, this child is having a severe periodontitis, which actually we do not see in children. We do not see cases of periodontitis, and there is deep pockets and severe periodontitis in a very little child. So this is an example of a leukocyte adhesion deficiency syndrome, which results from a missing receptor on the surface of the neutrophil, which does not allow the proper chemotaxis event to take place, so no adequate number of neutrophils can aggregate and accumulate at the site of the infection. Eosinophils are the other type of the granulocytes that have the eosinophilic part of the granules, and the eosinophils are really important in case of parasitic infections, and also can be seen in cases of allergies. So usually they do not participate a lot in the bacterial infections. They are more of a specialized form of cells, work at particular conditions of certain conditions of infection. Mast cells and basophils, both cells, they are different types of cells, as we mentioned earlier, but they share many features. Both of them contain histamine, and both of them has high affinity receptors for immunoglobulin E. So in case of allergy, mast cells and basophils are really active, and they would release a huge amount of histamine. These cells are activated by anaphylatoxins. So C3A, C4A, C5A, remember these complement proteins, these are the ones that are, once the C3 is activated, the C3B would go into the cascade, and C3A would work as opsonins, and it will also work as an activator for a mast cell and a basophil, and this is why we call them anaphylatoxin, because they activate the sort of an anaphylactic reaction. So these cells are responsible for the allergic reaction, whether just a mild allergy, just mild erythema, or anaphylactic reaction. This is mediated by the mast and basophils. Okay, so far we've been talking about these cells, mast cells. We talked earlier about dendritic macrophages, natural killer cells, humoral response. We talked about the complement proteins, and we talked also about the neutrophils and other granulocytes. These are part of the innate immunity. In the adaptive immunity, we have different types of cells. We have the B cells, we have the T cells, and we have now the natural killer T cells and the gamma delta T cells. They are still T cells. They are different than the natural killer cell that we have discussed today as part of the innate immunity. And maybe I've mentioned that I do not prefer the natural killer T cell. I prefer the cytotoxic type of T cells. This might be sort of variation of the CD8 cells with different receptors. This would be discussed later, but it would be confusing to call it natural killer T cell, because this is different than the natural killer cell that we have discussed today. So although we separate that there is an innate immune response and there is an adaptive immune response, but in real life this is not true. Both of them take place at the same time, and both of them interact together. It's like an orchestra or a symphony that each instrument is working separately, but if you want to hear the music, you need to hear all the instruments working together. So we cannot just, the examples that I have discussed, showing the migration of the neutrophils, the presentation of the antigen using the macrophages, and the presence of the complement proteins. This is not the whole story. This is not the whole picture. This is just part of it. I'm shedding the light on the innate part of the immune system. In the coming two lectures, we will be talking about the adaptive immune response, and then we will see how both of them work together, both at the same time. Of course, there is sort of maybe a time lag, like of course the innate immunity, particularly the physical and chemical barriers, would be the first line of defense, and macrophages and specialized macrophages located at certain parts of the body would be at the first line to face the antigen, but still the T and B cells would be able to conduct the immune function soon enough to catch up and to have their part in the immune reaction. So for the rest, the natural killer cells, macrophages, dendritic cells, and actually all these cells, particularly those three, would interact directly with the B cells and T cells to stimulate them and to create the adaptive immune response. So it is just to make things easier, we study each part separately, but at the end, when we finish studying each part, we will know how the whole process takes place. I like this cartoon. It just presents the difference between the innate immunity and the adaptive immunity. So this is here the innate immune response. There is no specificity. It's just recognizing certain patterns. So this is the menu for the innate immunity. This is what you have for breakfast, for lunch, for dinner. It's the same. There is no differences. But we will see, and in the coming 2 lectures, when we study the adaptive immune response, you'll see how you'll start mix and match things, how you would start having different options when an immune response is triggered or initiated, and why the first attack is different than the first exposure is different than the second exposure. So we will start to appreciate these things when we study the adaptive immune response. So the coming 2 lectures would be with so much mix and match things, and so much options are present that we will see in the coming 2 lectures. And by this we would finish the innate immune response, and we'll see you next week, start talking about the adaptive immune response. Thank you. Take care. Bye-bye.